All right, thank you. All right, I am going to introduce Dr. Marina Ramon. I've had the pleasure of working with Marina closely when she was faculty at Cabrillo College and managing the Title III STEM grant. She's also had a career working with SACNAS, doing research at the U.S. Department of Agriculture. She did her postdoc at the University of Southern California. And she's a board member at the National Postdoctoral Association. And now Marina works at a startup, Switchback Systems, as a scientist and project manager. We're super excited to have her join us today. Tell us about her career trajectory, how she got where she is, and learn about some of the research she's conducted in her career. So thank you, Marina, and I'll let you take it from here.
Awesome. Thank you so much, Sarah. Okay, let me share my screen. Make sure this is, I'm getting two different. Can you just see my slide? Yes. And now? Perfect. Yay. Everything will not go smoothly. All right. My name is Dr. Marina Ramon, and I currently work at a startup biotech company. And I think for the most part, this talk is going to, what I hope that you get out of this talk is that as you go through your career in science, recognize that it's okay to pivot and change in whatever direction. And sometimes things change in ways that you had no idea, and the skills that you learn at a particular job or whatnot translate to lots of other different jobs. And so I think the one thing I want to sort of hope you take away is that, one, don't be afraid to take chances. I think that especially if you are like thinking about, oh, should I really try this course? I don't know, like, or should I take this job? But it's really not what I want to sort of do. But you can maybe sort of think about what kind of skills that you will learn from that particular job and how that will translate to another job in the future. And you also the importance of networking. I can't sort of stress that enough. I mean, I am currently at my current job because of somebody I worked with 2 jobs ago. So, yeah, again, those are, I think, what I hope you get away, you take away with at this particular talk. I started out wanting to be a marine biologist, and I'll sort of talk about that. And now I'm sort of doing something completely different. And so when I started, I was really young when I decided that I wanted to be a marine biologist. I grew up in the Central Valley in Fresno, so it's pretty far from the ocean. So to decide that I wanted to be a biologist is kind of crazy. My family, my parents are from Mexico and came to the US when they were very young. And we grew up in the Central Valley. I have 3 brothers, and my family's pretty traditional. I was the only daughter. I was supposed to be the one who took care of everything and made sure that dinner was cooked, things were cleaned, laundry was done, all those sorts of things. So, I think the one thing, though, that my parents did really do stress throughout my life was education. My mother had a 6th grade education and my dad graduated from high school. So they understood that the only way I could sort of move up economically was through education. That was sort of their biggest push. So they were constantly pushing me to get a better education. And when I talk about taking chances, and especially in education, sometimes it can sort of be really hard. I went to a school that was 99.9% free lunch. So pretty much everybody had free lunch, and came from a neighborhood that was not the best neighborhood in town, for sure. There was few who were educated, few who had jobs. And for the most part, it was tough. And, there's a lot of, violence in general, like drive-by shootings, like domestic, but it was a tough neighborhood that I grew up in. And so I think that my parents, again, one of the reasons why they stressed education is they could have understood that that was going to be my way out of there. So early on, when I was in junior high, I was accepted to a magnet school it was a science and computer magnet school. And I really did like science a lot. And it was there that I sort of learned about like what a marine biologist does. And we went to the Monterey Bay Aquarium and I met this woman who was a scientist. And it was the first time I'd ever, one, met a scientist, two, met a woman who was a scientist. So I just remember thinking, whoa, if she can be a scientist, then I can definitely be a scientist too. So I think that was sort of eye-opening. And, and I went to a school that magnet school had a lot of students who were very affluent. And so when we talked about like, what college are you going to? Most of them were like, I'm going to Harvard, I'm going to Stanford, I'm going to this place and that, that's where my parents went. And I just remember thinking, holy smokes, like, I can't believe these students are going to go to these colleges. Like, that's crazy. I only have heard of them like from TV or something. So, we'd have college recruiters who would come. And I remember talking to a student who was right next to me, and he turned and said, Marina, what school are you going to go to? And I was like, I don't know what college. I'm in 7th grade. I'm 12 years old. I have no idea what college I'm going to go to. And he's like, oh, well, I'm definitely going to Berkeley. And what really like struck me was that one, he was like a C student. And he was like, oh yeah, I'm totally going to Berkeley. And I was like, well, I'm an A student. If he's going to get into Berkeley, then I can get into Berkeley too, like no problem. So I think it was sort of those like little messages and things that helped me sort of get the reinforcement and the confidence to be like, okay, if this person who I think is sort of, the same level of education or, of smartness, I guess, I don't even know, or then if they can get there, then I can get there too. So I sort of was dead set, like, I am going to be a marine biologist. I'm going to go to UC Berkeley. Like, that's what's going to happen. And I ended up getting accepted to Berkeley, but then decided that Berkeley wasn't for me. I decided to go to UCSC, and I still decided that I was going to be a marine biologist. Like, I was dead set, like, I want to be a marine biologist, and I think trying to navigate higher education is really difficult when you don't have family or anyone to sort of help you guide you through that. So, I had to, it was like back in the old days when you had to like on paper fill out your UC application and like get a waiver. I mean, it was really, now it's like everything's online, you do all this sort of stuff, but back then it was like on paper, and I couldn't just like Google or look on the internet like what I wanted to do. Instead, I had to ask a lot of questions, and had to ask for a lot of help. That was the one thing that I can't stress enough, is that even when you think you know the answer, you should still ask, and ask for help often. And don't be embarrassed. I had parents who barely had a high school education and junior high education. And I would just tell people like, I don't know what this process is. Can you please help me? And everyone is super wanting to help and make sure that you're successful, like, especially in academia, like people, teachers, mentors, everyone really, and counselors really want you to succeed. So don't be afraid to ask. And if someone didn't know the answer, then I would go ask somebody else. Like, they're like, oh, I don't know that. And I'd be like, well, is there somebody you can send me to? And I would go and ask as many questions and get as much help as I could possibly get. So I ended up going to UC Santa Cruz. And it was a great place for me because it was a smaller one of the smaller universities, one of the smaller UCs, and I tried to take advantage of as many programs that could help me be successful. I think one of the programs that I worked with was called ACE. It's an Academic Excellence Program. You know, they support students in making sure they're successful in the courses that they take. They have extra tutoring. I ended up being an ACE tutor. Again, like I said, I can't stress enough, like if you're struggling or you need help, find a group, ask for help. And you know, what I loved most about ACE was that it was a small community. It was a community of students who were all in the same place, and we were all trying to be successful. And so we all helped each other, and we worked together, we studied together. And sort of, I helped create that sort of family around me so that I could be successful in making sure that I was going to pass these courses. And let me tell you, there are courses I did not pass. I mean, so even if you fail, don't give up. You know, I got a bachelor's degree in marine biology, but it ends up that I was really interested in genetics. And I failed genetics at UC Santa Cruz. I failed that course miserably. And I remember thinking, oh my gosh, how am I going to be a scientist if I can't even pass this like my first upper division graduate course, you know, undergraduate course. And then I took it this second semester, and I remember hating genetics, thinking this is like the worst class ever. I don't ever want to take it again. Took it with another professor and ended up loving everything about it. I just remember thinking, wow, this is actually the most interesting stuff I've ever learned. And this is, I don't want to be a marine biologist. I want to be a geneticist. But before that, I sort of had to also make sure, is that really true? Do I not want to be a marine biologist? So I did some internships. And that's the other thing I recommend too, is internships. I did an international research training internship in Argentina, and I was in Patagonia working on southern elephant seals. That's what this sort of upper picture is. Those are elephant seals that we tagged. You know, they go through sleep apnea, so you can basically like push them and shove them and they're not going to wake up. And we would tag them and keep track of them during their molting season. And I spent 3 months doing field work in the middle of nowhere. Electricity didn't get turned on until like 6 o'clock in the evening, and it was only on from 6 to midnight. And it was some pretty harsh conditions. The closest town was 2.5 hours away. We only had communication with them via satellite phones. So it was pretty remote. And it was at that time that I realized, although I love marine biology, I don't like this kind of field work. Like, this is not for me. Like, I am like, it just wasn't the type of work that I like to do. And I really thought that was what I wanted to do. And so I had to, you know, rethink about like, what is it that I want to do in marine biology? And one for sure is I did not want to work on marine mammals, and I didn't want to do that much field work. I like the field work, but it's not like roughing it for 3 months in the middle of nowhere was really tough. So then I did another internship. Like I mentioned, I really loved genetics. After I had failed it, I loved it the second time around. And the professor who taught the course had a second course that was called population genetics. And I remember taking his course and thinking, oh man, this is like the coolest stuff ever. And he said, oh, I need a couple of volunteers to help us in the lab, like a couple of hours a week. And I just remember going, yes, 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 please, I'll do whatever you want. Like, I'll totally do that. And he worked on Drosophila. That's what these little flies. I don't know if you ever have like dead, you know, bananas that are rotting. That's what I worked on, those guys. And you can notice that they have red eyes and white eyes. White eyes is the wild type, and red eyes is a mutation that occurs. Or maybe I have it the other way around, but in any case, we looked at mutations that had happened, and he was really into sort of population genetics, and I volunteered in his lab. And then I heard about an internship called MARC. It's Maximizing Access to Research Training. And part of the MARC program is that you find somebody who's willing to sponsor you, and they will pay you for the summer to work in their lab. And then they'll give the PI, or the principal investigator, that professor, some money so that they can let you do research in their lab. So I asked him, hey, you know, I really am interested in doing this internship, but I need to find an advisor. Do you think you would do it for me? And he's like, oh, absolutely. Like totally, you know, I mean, it was really scary to like ask him like to do that. But again, if you don't ask, you don't know whether or not you can do it or not. And if they say no, that's the worst that will happen. It's like, no, not a big deal. And so I spent a summer counting these little flies, which was tedious. And although I loved population genetics, I knew that I was never going to count another fly after that summer. Like, it was like, I spent months and months and hours, like counting flies, like putting hundreds of flies. Like, so I was like, no. Although I love this project, I don't like doing this type of work. And I like the lab work, but not this kind of lab work. So it took me, you know, doing these internships to help me realize, like, really narrow down what it was that I wanted to sort of do. And along that way, the principal investigator, the PI that I worked with, that worked on the fruit flies, he sort of really helped me recognize that I could be a scientist. I think even then, when I was young, I had sort of this imposter syndrome of thinking, I don't know if I really belong here. I don't know if this is for me. I mean, everyone says, yes, you can do it, but I'm still not sure I can do it. And I remember we had our first meeting, and he told me, you know, Marina, when I want you to write this grant proposal for the summer. That's going to be your project. I want you to write a proposal. You know, you're going to need this skill set, especially, you know, after you get your PhD and you become a professor at a university. Like, this is something you're going to have to do for the rest of your life. And then he said a bunch of other, like, blah, blah, blah. Because all I remember him saying is like, whoa, he thinks I can be a professor and a scientist. He thinks I can get a PhD. I remember thinking, wow, holy smokes, like, I don't even think I can do that, but he thinks I can do it. So maybe he's right. Like, I'm not sure. So I just, I went with it. You know, in times of doubt, I remember thinking, wait, Bill said I can have a PhD, and he thinks I can be a professor and I have the potential to do that. Like, then, I mean, he's like a famous scientist. Like, he has all these papers. He's done all this research. He's done all this stuff. Like, if he thinks that, then he can't be wrong, right? And so I think, you know, I took that lesson, and when times of doubt, I remember thinking, wait, Bill said I can do it. That means I can do it. So after I finished my work at UC Santa Cruz and got my bachelor's degree, I ended up getting a master's degree from Boston University. And that's when I finally sort of really helped me narrow and realize what I want to do. Yes, I wanted to work on marine organisms, just not marine mammals. And yes, I still wanted to do the population genetics and in that genetics work, because that evolutionary work, because that's what I was really interested in. So for my master's degree, I spent time working on these fish. These fish are called hamlets, and they're simultaneous hermaphrodites. That means that each individual is a male and a female. And so, you know, I was really interested in, like, well, how are species formed? Like, how do they know who to mate with if they all, it just seems sort of interesting to me, because morphologically, they're all about the same size fish. They have this, you know, most of the time when species are identified, they're based on, like, how many ray fins do they have? How many gill sets do they have? You know, that sort of thing, like what color, what's the depth, what's the length of all these different things. And all of those characteristics are pretty much exactly the same for these fish. The only difference between them is their color morphology. That is what's different between these fish. Otherwise, you can't, other morphological characters, they're all the same. And so I was really interested in understanding, like, how are these species being maintained just because of that? You know, in the wild, they have positive assortative mating, which means that one fish who is yellow and with a blue sort of snout will find another yellow fish with a blue yellow snout, and they will come together and they will swap eggs and sperm. One will act as the female and the other one will be the male. And then they'll share eggs and sperm, and then they'll switch places, and the other one will be the female and the other one will be the male. So they do sort of this swapping of eggs and sperm. And so I was interested in using molecular markers. At the time, scientists were using DNA, and they were using mitochondrial DNA. Mitochondrial DNA is inherited from your mother. And there's several genes. It's a circular genome. It's only found in the mitochondria. It's only shared from your mother. So only you and your mother have that same copies of those particular genes. They're highly conserved, and there are regions that are not conserved. And so they use those markers because they know what genes are there, so that you can sort of sequence and look at the DNA of a particular gene and then compare it with another individual. And let's see. Oh, that's weird. I'm not sure why that happened, but the lessons that I learned from, so I'm not sure what happened, but while I was there at Boston University, I also was asked to run their sequencing facility. The person who I worked with was my co-PI. I had a PI when I started, and recognized that one, he didn't have a DNA lab. He didn't have a lab that I could even extract DNA in or do any of that sort of work, any of that molecular work. So it ended up that I had to go find somebody that I could do that. He had all the samples. He knew all the knowledge about the fish. I mean, he was a true ichthyologist. That's what he sort of did. But he didn't do any of this molecular work. And so I had to go find somebody. So I went and sent emails. I knocked on people's door and said, hey, and he gave me the names of a couple of professors that I thought I might be able to help me out. And I had to, again, go and ask, like, I'm new here. I have this project. Can I work in your lab? Like, I have all the money to do it. Like, I have everything to do it. And so I ended up working with a really great professor. His name's Michael Sorensen. And at the time, he also was in charge of their sequencing facility. And so after about a few months of working with him, he asked if I could run their sequencing facility. And so I did. And I did it because I thought that, one, it would be a good experience for me. I mean, it would be a good experience for me to like run a lab. I mean, if I'm going to be a professor running my own lab, then running a sequencing facility would actually be good. I have to deal with budgets. I have to deal with ordering. I had to train students. I had to train professors and faculty members. I had to run samples. So I was in charge of like doing all of those things. And I figured I had nothing to lose and everything to gain to sort of do it. And it would also give me the skills to run my own lab. And so I did that. And I was really, I mean, that was my biggest lesson, is that one, I was kind of like, how am I going to be a student and also now full-time manager? It was tough, but I think that in the end, it was worth it, because again, the skills that I learned from that particular position helped me in the future and many other positions. And with regard to my research, it ends up that species and how new species are formed is super complicated. It wasn't such a simple answer. Like, I thought, oh, okay, like these fish, based on their color morphology, they mate with each other. That means all the blue fish will be closely related based on their DNA. All the yellow fish will be closely related to each other based on their DNA. And it ends up that that's like, yeah, no, it's super complicated than that. And in that sort of process, because I worked on the mitochondrial DNA, this is sort of the mitochondrial genome. It's a round genome. I don't know if you can see my arrow, can you? Okay. It's a round genome. And each of these purple and yellow sort of portions are different genes. And like I mentioned before, it's highly conserved. All mammals and pretty much all vertebrates have the same gene order. There's lots of, they have the same gene order in general. They have also a region called a control region, which is found, it's typically around here, this sort of region that's sort of blank. That's the control region, and that's the origin of replication. So when the mitochondrial genome is replicated, that's where it begins to unwind the DNA, and it's replicated and you make a second mitochondrial genome. And so it ends up that these fish actually have a gene rearrangement. which at the time was completely unheard of. When we originally brought up, when we started sequencing and we couldn't get things to work because we were like, wait, these primers based on where we think the gene is should work, and we couldn't get things to work. It was super frustrating. We spent, or at least I spent, like months and months going, what on earth is sort of going on? And so very early on, we realized, I think we thought it was a gene rearrangement, but everyone was like, no, that's impossible. Vertebrates have highly conserved genomes. There's no way, like only like snakes and like birds have these different sort of genomes and whatnot. And what ended up happening is that I ended up sequencing the entire genome. And it's about 16,000 base pairs. So each base is like in DNA is either an A, C, G, T, each of the different nucleotides. And it was painstakingly sort of slow because it was old technology, like this old sequencing technology that we don't even do now. It doesn't even happen now. Like what I did back then was like pour these huge gels and like ran it for like 16 hours. We got each of these bases one at a time. It was quite cumbersome, but now it's, you have these like new sequencers that are basically like this big and plugs into your computer, and you can run your own sequence like on your on your computer in your science lab. But before, you had to have this huge facility and did all this sort of stuff. So yeah, it ended up that speciation was complicated, but in that process, we ended up discovering something else. And like I said, it's complicated because for these particular fish, when we talk about speciation, we're talking about an evolutionary timescale, which means that right now at this time is just one time point in this evolutionary scale of how things are differentiating. So when we talk about species, we usually think of this is species number 1 and this is species number 2, right? And then sometime long ago, if you go back and you trace back their DNA, they were all related to this particular individual. That's if we capture speciation at the endpoints. What these particular fish was happening is something different, where they're in the middle, they're at the very beginning. They're right here at this axis, this point, at this fork, where maybe in the future they might become a full species, 1 and 2, where if you sequence particular genes in their genome, they're absolutely different species. But right now at this evolutionary time point, they're not quite there yet. And so it was an interesting lesson for me to learn, because when I think about it, I think of like, this is species number 1, this is species number 2, this is what people called it. And in fact, that's not true. Like, you could have species that are emerging and they're just emerging, and we can't quite differentiate them. They are just starting to begin to show the traits of being different species. And that's what we ended up finding for these individuals. So it really got me going, like thinking about like, wow, wait a second. That means that there must be genes involved in speciation. I mean, right? Like when we think about how species work, we think about molecular evolution. We think that you have some DNA, somehow your DNA in the environment, the environment influences your DNA, and then you become sort of this organism or this species based on your environment and based on your DNA. You know, this is sort of a classic dogma of molecular evolution, where you have a genotype, which is your DNA, your environment, and it equals the phenotype, or what the organism sort of looks like, the characteristics of an organism. So I spent my entire PhD and my postdoctoral research really thinking about like molecular evolution. How are species related to each other? How does gene flow influence populations, and how do at some point they become different species? You can have one species that has an entire spans an entire, like 2 mountains or something, or spans an entire range or area. And then eventually at some point they become 2 different species. It'll be 1 species at once, and then something happens and it'll become 2 species. So how does that influence? I was interested in that, and what types of historical or biological factors could have contributed to their current structure, population structure. So that's what I spent my PhD work and my postdoctoral work. And it's a lot of work. I ended up focusing on fish because fish are, 1, easy to work with. They're all over the world. You can go anywhere in the world and decide to work on those particular fish. And it ends up that I worked on fish that were intertidal fish that you find here in California. So if you go to the tide pools off of anywhere in the Central Coast, or even Northern California, Southern California, all the way to Mexico to Alaska, you will find these little tiny fish, and they're called sculpins. And I spent my entire PhD and postdoc working on that. My PhD, took me like 7 years to finish my PhD. And that wasn't because I wasn't working hard. I was working super hard, but I was also working on a lot of other things. That was what I ended up sort of doing. And again, like I said, I felt like my PhD time was, yes, I wanted to get my PhD and finish a particular project, but I also wanted to grow professionally as a scientist. Like I wanted to make sure that I was taking lessons perhaps from other ecological systems and thinking about how does that translate to the current system I'm working, like thinking about how other scientists do their work. How can I use that knowledge to work on my particular work? And different tools, thinking about all the sort of different tools that are out there. Can I use those tools to answer my questions about, like, how are species created? How are populations connected to each other? How are they influenced? How are genes influenced? Those sorts of things. One of the things I did at the very beginning of my graduate work was I took a course. Here we go. I took a course and spent 3 months in Costa Rica working on experimental design. And so I learned how to quickly, like, come up with a project, come up with an experimental design, implement it, and do it. And it was probably the best experience that I could ever have had, in the sense of really teaching me how to come up with a question and actually implement the research and do it. And that's a hard thing to teach, I think. And so that course was great. I worked from working on bats to butterflies. I mean, I worked on everything. It was awesome. And so I did that. And also I worked on several other projects while I was in my PhD that we ended up publishing papers on. I worked on clownfish, those, Finding Nemo. I'm sure you guys have all heard of Finding Nemo. Those little clownfish, I worked on those little guys. And they're super easy to catch underwater. Well, they're sort of easy to catch. You sort of throw a net over an enemy, you catch them, and then we would catch them and then fin clip. We take a little clipping of their fin so we could have a DNA sample, and we put it in a tube and then release the fish back to his anemone. So it was a capture and release. So lots of scuba diving, lots of collecting fish in some nice warm waters. It was awesome. And then I also taught a course while I was at grad school. That was the other thing. I had the opportunity to teach a field course, which was super fun. So that was really great. And then I took courses on, how to raise fish, how to actually look at morphological characters of fish using different kind of staining things. So again, like I said, I tried to learn as many tools as possible to help me answer the question that I was trying to answer. And during that time, I also did it was like a 3-year fellowship. It was called CILS. It was the Center for Innovative Learning and Science. And it was a collaboration with UC Santa Cruz, the San Francisco, oh my gosh, I can't think of the name of the science museum up in San Francisco, Exploratorium. the Exploratorium, and then King's College in London. And they were looking to help teach scientists teach science. So they were trying to, there was a new program. The idea was, how do you teach scientists who are like focused on just research? How do you give them the skills so that they can teach science to other students and novice scientists? And so we had to sort of do this sort of project. It was a 3-year program, and I spent time doing that. And one of my project was, it's an ongoing project that has been going, I think, on for the last like 20 years or so. It's called, maybe it's even more than 20 years old. It's called Draw a Scientist. So you give young kids a blank piece of paper and you tell them, draw a scientist. And the idea is that hopefully over time, we really break down and change what students think and imagine the stereotypes of what scientists look like. So for example, at the very beginning, when I would meet with kids, they didn't know I was a scientist. They had no idea what. I would go to their classroom. I'd be like, oh, I'm here to visit and I have this great project. We're going to draw a scientist today. And they would draw scientists, and it was typically this like mad scientist, bald with crazy hair. Like they all had these lab coats. They were blowing up stuff. Like I would say 90% of the students drew scientists as that. And then I would go back and I would tell them, oh, actually, I'm a scientist and this is the research I do. And I talked to them about what it's like to be a scientist and things like that. And then we'd have them to draw a scientist again, like after they had met me. And students actually drew realistic scientists, like what, usually was pictures of themselves being a scientist or pictures of scientists doing like real work, something, So that was super fun to do. And I think it also helped me think about like teaching, because at some point, if I wanted to be a research scientist at a university, because that was sort of my plan, I was going to be a professor and have my own lab, then I needed to know how to teach. So that's part of the reason why I did that. And so after I graduated, I actually got a pretty prestigious award. It was an NSF, it used to be called a Minor Postdoctoral Fellowship. I think now it has a different name, but at the time that's what it was. They give out, I think, 4 or 5 postdoc fellowships to anyone, any new like PhD student who writes a proposal and whatnot. And I got it. And it was to work at USC and to continue my PhD work. And again, we had during my PhD, I had discovered that the fish that I worked on had a little bit of population structure. They were, again, these budding species that were at the very beginning of becoming new species. It was we knew that there was genes that were involved that differentiated different populations. Because these fish live in small tide pools, they're typically, I'm running out of time here, so I'm gonna like go super quick. Anyways, I spent a lot of time working on these particular fish, and I ended up, again, learning a new tool. I worked on something called microarrays. Each little spot is a type of, it's DNA on, that is a spot of DNA. And then we use other DNA, and then we can sort of, they bind together, the different single strands bind together, and then we can look at the data and determine like which genes are being turned on and off during, for this particular example was hypoxic conditions, because we knew that these fish were under hypoxic conditions at times. And during my postdoc, it was during the recession. And so it was tough, like finding a job. And so I had to rethink, what am I going to do? Like there was tons of academic positions that were being pulled, budgets were being cut, there were no jobs. It was like grim and bleak. And so I decided that I was going to work for a nonprofit that I had once been a member of, and still am a member of too. And it was called SOCNAS, the Society for the Advancement of Chicanos and Native American Science. And I was one of their program managers there. I did everything from running the conference and actually putting a conference together to creating new programs for professional scientists. And what I loved most about working for this nonprofit was the fact that you end up learning, you become a jack of all trades. You learn all kinds of things. Like I had no idea what ROI is, return on investment. I'm like, when someone say, oh, what's the ROI on that? I'm like, I don't even know what you're talking about. You're like talking a foreign language to me. Like, and so I learned all these sort of other skills. I learned how to communicate with people. I learned how to fundraise. I learned how to bring teams together to get something done and be at this mission. Even though, I mean, I'd done that before as a scientist, this was something completely different. And so again, like tools and skills that I learned at this particular job is what has helped me at the job I'm currently at. But what I learned, and the other thing I loved best about working at a nonprofit is that the mission, everybody has the same vision and mission and are so dedicated to doing it. Like I love that passion. But at the same time, you work crazy hard and it's really exhausting and tiring. And at some point I was like, okay, I need to go back to science. Like I can't do this. And this again is where networking helps. You know, I had networked with tons of individuals and scientists, and a position came up at USDA to work there. And I thought, and I knew somebody who had worked there, and I was like, oh, I think I could sort of do this. I can go back to science, even though I've stepped away for a few years. I'm not that far away, and I still know some of the current science that, you know, especially around sequencing. Even though we're doing new things, I have the skill set to be able to learn those new things. And so at USDA, we used a molecular tool to detect soil-borne pathogens. And these pathogens, you know, I mean, they just like kill these, especially strawberry plants. I worked on macrophamina, which is called charcoal rot. And that just kills crops and crops of strawberries. And as you can imagine, here in Santa Cruz or in Watsonville, Salinas, strawberries are an important crop in the Central Valley. So people or Central Coast, and people are really interested and want to make sure that they are on top of that. So we used PCR, qPCR, we used all kinds of different tools, molecular tools, mainly looking at DNA to understand how we ran population genetic studies. We understand how these soil-borne pathogens, how are they related to each other? If I collect one in Salinas, is it related to one in San Luis Obispo? You know, so we use those particular tools to sort of ID and establish different species. And while I was at USDA, I also sat on their environmental hazard and safety committee. You know, it's one of the things that you sort of have to participate in the committee. And I'm like, well, I should learn this. Like, this is important stuff. I work with sometimes some nasty chemicals. I should learn this stuff. And then I also continue to sort of do the outreach work that I did. But it was in Salinas, and it was pretty far in a commute. At the time I had small kids. So I had to really, really think like, can I continue living like this, like spending 2 hours a day in traffic and, you know, miss out on my kids? And so I ended up finding a job at Cabrillo College. And again, networking. I knew people who worked at Cabrillo College, and somebody had emailed me and said, hey, Marina, I know you want to work closer to home. I heard there's this job. I think you would be good for it. And I remember thinking, sure, why not? I'll apply. And I ended up being the director for USDE Department of Education, Hispanic Serving Institute STEM director. We did a whole bunch of stuff at Cabrillo and lots of project management work that was super fun to do. We built a greenhouse with Sarah's help. I mean, Sarah did most of the work. All I did was like, okay, make sure that things got paid. And so with Wendy, like get things paid. And we did all that sort of like back work, which is a ton of work too. But it definitely was super fun to do. And again, working on a team, getting things done. And then I also was, and I still am, an associate faculty, and I teach, sometimes I teach a microbiology and an applied research course at Cabrillo. But once the grant ended, I was thinking, which direction should I go? Should I go back to science or should I stay in academia? And I was really interested in going back to science. So a friend of mine who had worked at, I worked at USDA, moved to a biotech company. And this biotech company is called Switchback Systems. And again, I've sort of gone all over the place. So like I said before, the skills that you learn along the way will help you get to your next position, and always think of them that way and take them that way. I think that my title here is scientist and project manager, and it's because I had that project manager skills before. I had been on a committee for environmental hazardous waste and safety committee. Like, they needed somebody who had all of those sort of skill sets and sort of also knew the science. So that's part of the reason why sort of I came here, because it was also, we're at a time where in this biological revolution where we really do have the ability to read and write DNA, which is sort of the foundation that's going to get us to the next steps. And it's just a matter of like, how fast can we do that? And our company is working on creating a DNA synthesizers using chemical synthesis. I'm going to go through this super quick because we don't have too much time, and I want to leave some time for questions. But what the plan is that when we think about DNA that is created, we go back again. I talked about this before, that DNA is made up of A, C, T, G. These are the building blocks, the nucleotides of DNA. And then you sort of build it together and you can make an oligonucleotide, and then a gene fragment. And then you sort of have genes and chromosomes, and sort of this is sort of how we think about like the sizes of the DNA. Each, depending on the size, it has a different name. And one of the problems that we have is that oligos are super small. They're great. They're super easy to use, but loss of privacy, lack of control. And as these DNA samples get larger and larger, you have more and more problems with regard to customer. So before, I was on the customer side. I was the one who ordered the oligos or had oligo pools and things like that, or wanted gene fragments. Now we're on the side of making those. And again, different industries use them for different reasons. Vaccine development, personalized medicine, life science industry, as well as the government. So there is a need out there, and there are particular frustrations for each of these different industries. And the goal is that what we will do is sort of help deal with all of those frustrations. There's several companies that are out there, and ours is sort of an outlier of what we want to do, because we're actually going to build the instrument so that the customer can make their own DNA, as opposed to having somebody else do it for you. I'm going to skip through that. Again, the instrument is going to be quick turnaround. You have control of it. It's privacy. It's easy to use. It has high yields, very little waste. And not only will you be able to make you can make one strand or multiple strands of DNA, so all at once. And so it'll be, you'll be able to assemble things at a much faster, at a bigger scale and faster and much more flexible. Right now, we are working on our total access, our tech access program. We're trying to get oligos to customers. We're working on doing that. We will have an alpha in 2023, and at some point in 2024, we'll have a beta machine ready to go. We had pre-seed money. We are currently on our seed money, and at some point we will work on seed 2 money. So that's where I'm at. Thank you guys so much. If you have any questions, I'm going to stop sharing so I can see you guys. And there.
Thank you so much, Marina. Let's see. Does anyone have any questions? Feel free to unmute and speak freely, or drop them in the chat and I can read them out loud.
Please ask me some questions.
I'll ask one while other people kind of get their brains around their questions. So Marina, most of your life and professional career was in California, but you did that one master's a couple of years in Boston. And could you talk about just how that was for you, if any students are thinking about transferring to schools far away, or maybe what to keep in mind?
Scary. That's definitely what it was at the very beginning. I have no family, knew nobody, but I knew that it was something that I had to do in order to better educate myself, and also sort of have a better understanding of what the world is like and living somewhere else and on your own. I think that that was the one thing that I've accomplished that I'm most proud of, is literally packing my bags, having 2 bags, getting on a plane, having nowhere to live, because again, back then there was no internet. You couldn't really sort of, I had to live in a motel for like 2 weeks until I found somewhere to live. And it was scary. And I did it by myself. Like, it was kind of crazy, but I think that there's other people who have done scarier things than me, and have done things on their own and don't recognize how brave you are to do it. So, yeah. So kudos to everyone who sort of has done that, and recognizing that it's hard and it can be scary, but in the end, it's super rewarding, and you shouldn't be afraid or let that be a limitation to what you want to do.
Does anyone else have a question they'd like to ask Marina? Marina, what's your favorite organism you've worked with?
Oh, well, as you can imagine, I love fish because they're so awesome. And I think I love them because if you look at the evolution and the adaptation for fish, I mean, they live in the most hottest and the most coldest places in the world, and everywhere in between. There are fish that can literally walk out of water, and especially these intertidal fish, they can walk out of water and go to another pool if there's not enough oxygen in the pool they're in. I mean, I think that for me as a molecular evolutionary biologist, like, that to me is just fascinating. Like, that that's all. I mean, that's amazing. You have simultaneous hermaphrodites. Oh my gosh, you're both a male and female, and you mate with another individual. Yeah, fish are by far the best. I mean, I'm sure there's other people have other biases, but fish is mine.
We have time for another question or two if anyone else has one.
You know what I forgot to mention was the other thing too, is that, even though my family was super supportive, they were supportive in me getting a bachelor's degree. And then at some point they said, wait, Marina, you need to get married and have kids. Like, what's up? You got a degree. Be done with it already. Like, come on. Even though they were probably not as supportive and gave me like some pushback, I knew that this is the direction I needed to go, and it was what I was going to do. So even when my close family were like going, no, you need to go get a job and do this and go get married and have kids, and like, and really laid on the thick guilt trips. Like, I still was like, no, this is what I'm doing. Like, I know that this is going to be the best move for me. So yeah, I still had to go. There were several times that I was totally had to go against my family. And even my husband, who was my boyfriend at the time, was like, what, you're moving to Boston? I'm like, yep. Like, I'm still doing it. Like, so I think that you should go with your gut feeling, what you know is best for yourself. Even sometimes when you have plenty of supporters who are, I'm sure, super happy, but there'll be sometimes when they don't agree with what you're doing, but you know what's best for yourself. You should still do it. They'll come along. They always do.
Thank you, Marina, for adding that in. And we do have a question. do you think it would be better to find internships in the field you're looking at? Or would choosing something a little far from it offer a more rounded experience? Thanks, Tiffany.
Yeah, that's a great question, Tiffany. You know, I really think that if I know several people who've done internships that are sort of outside of their field and have gained great experience from it, especially in science, when you're thinking about like just experimental design, how sort of things work. It sort of is super helpful to do any sort of internship. Because most of those internships really will sort of give you the foundation and skill set. And you also never know if you're sort of like, I kind of think I like that. Maybe you do it and you're like, holy smokes, I love this. This is what I want to do for the rest of my life. You don't know until you try it. I mean, that's the most I can sort of tell you. And then, you know, you could be in my position where I was like, I want to do this. This is what I want to do for the rest of my life. And then I did the internship and was like, oh, heck no. I don't want to spend the rest of my life doing this. Like, so I think that that's also, it's worth it to do what you can and make sure that you get your, get experience doing an internship in general. It will be a valuable experience and you can't go wrong.
One thing I've noticed, because now this is the fourth one of these I've hosted, it does seem like internships are like these really key moments in people's careers, because it is the first time you really get to dive in, you know, and practice something, figure out if you like it. So yeah, I think that's great advice. Thank you.
Yeah.
All right, it's after 6, so unless anyone else has a pressing question, I think we'll wrap it up. Let's give Dr. Marina Ramon a round of applause. Thank you. Awesome.
Thanks.
All right, anything else, anybody, before we kind of close up here? All right, thank you, everyone.
Bye. Thank you.
Thank you so much. Bye.